Hello, I'm John True, and this is my show and tell talking about my research. I apologize to the recent cohorts of E&E grad students who, I wasn't able to make that time slot for show and tell, and it took me a couple years, but I finally got around to actually making this. So now it's actually going to be up to date with what I'm doing these days. So I'm interested in how phenotypic diversity evolves, and I work on morphological traits, especially pigmentation. I'm also really interested in behavior and its evolution and genetics, and also the genetic underpinnings of speciation. For approaches, I use the Drosophila model system. So we have a lot of classical genetic tools. I've done some developmental genetics in that system, and that has informed the evolutionary genetics approach that I use. I've also used QTL mapping and also computer simulations. We studied some aspects of speciation with simulations, and as I'll talk about at the end, I'm starting to study behavioral evolution using computer simulations. Drosophila elegans has been one of our main model species. It's a member of the Drosophila melanogaster species group. It's endemic to South and East Asia, and it breeds on flowers. There's a picture of elegans on a morning glory in Taiwan. So it grows really well in the lab. We've done a lot with it, and it's interfertile with a sibling species, and so we've used that system to do some genetics to ask about the genetic architecture of pigmentation and behavioral trait divergence. So here's a movie I'm going to play, and hopefully it plays within the movie I'm making. If not, I have a link for this on YouTube. So that's an elegans male courting a female. The male is the one with the black spots, and you saw that it went out in front and did this little thing we call the courtship dance, in which it looks like it's displaying its spots to the female. So that's not typical of Drosophila melanogaster, which is a species that doesn't have male spots. And there's a bunch of male spotted species in that species group, and there's a lot of species that don't have male wing spots, and the ones with wing spots all tend to do some form of that display, and the ones that do not have wing spots do other things, but they don't do that going out in front of the female and displaying the wings. So for a video of that previous slide, if it didn't work within this video, there's the YouTube link. The sibling species is Drosophila guanicola. So far it's only been isolated from Indonesia, where it's sympatric with Drosophila elegans, and there's a male up above courting the female. You can see this species doesn't have spots, and it has a much more sedate courtship ritual. It pretty much does what you saw, and then the rest of it is just from behind the female attempting copulation. These two species are interfertile, and we've done the hybrid and F2 genetics and some QTL mapping that I'll talk about. The URL for that video is at that link right there. Here's a video of Drosophila elegans, which has a lot of different hosts in Taiwan, including several hosts that are not native to Taiwan. So here's one, Brazilian jasmine, it's from the New World. The morning glory is another one. We think its native hosts are hibiscus and wild ginger, alpinia, among others. But it's got a lot of interesting ecology on the flower. So what happens is the females will lay their eggs in these flowers that only last a short time, and the flowers will drop, and the larvae will feed on the decaying flower material. So hopefully you can see this video. This is a video. It's in an urban setting, right outside the apartment building we were living in, a garden.
Yeah, we've got Drosophila elegans courting.
The noise you hear in the video is street noise.
The flower
it's very easy to find the
Brazilian jasmine, which is an Apocynaceae family. I don't memorize the species name yet. See that.
Male is actively courting a female. Okay, so the URL for that is at that link right there. So we've actually been to Taiwan looking for these guys many times. We have collaborators over there. And we found it on a lot of different species in different places and different times of the year. So Hibiscus taiwanensis is a native hibiscus there. It's very common on that species. And several other hibiscus that we've seen there. Alpinia is a wild ginger. And especially up in mountain areas, it's found on that. Thunbergia erecta is this purple one down to the right. This one to the left here on the lower left is Alpinia. And then the one you just saw is Mandivula, the Brazilian jasmine. And we actually haven't fully characterized the flowers that Drosophila elegans does this on, but it's really a lek-like system. The males will go to the flowers, females will come, the males will court, the males will also have aggressive behavior towards each other. And it's really it's a unique ecology among Drosophila, and it's a really interesting ecology that hasn't been studied too much. So over the long term, we'd like to characterize all of the species that Drosophila elegans uses, because it's probably very interesting. They have some seasonality there, although it's the subtropics and tropics, and the yearly dynamics or the seasonal dynamics of that are probably very interesting. And also, there's a lot of deep-bodied flowers that it doesn't use. We'd like to know why it uses the ones it uses and why it doesn't use the other ones. So that's a long-term interest. We've done some experiments on this behavior in the lab. We have an ability to have flies that don't develop the spots. If you do, it takes about a day for the pigmentation to develop on the wings. If you cut the anterior wing veins with a pair of micro scissors, just as they emerge from the pupal case, then you can prevent the spots from coming in. So we did a lot of trials where we had 2 males and 1 female. 1 male which doesn't have the spots, 1 male which got the spots, and then we did a mock surgery. And then when you do that, you can see that in some cases there's no difference between the ability to copulate, so such as in this experiment. But if you look at the number of courtship bouts it takes for the male to get a successful copulation, then the non-spot ones, and this has been repeated a couple of different times, the non-spot ones have to do more courtship bouts to get a successful copulation. So we think that without spots it must be harder. It might be harder work, and so the spots might serve as a ornament to help stimulate the females. So, and we've done backcross genetics in this group. So the hybrid females between Drosophila elegans and Drosophila gonicola are fertile. The males are sterile, following Haldane's rule, which I'll talk about in a few slides. And this is a genetic map of the species pair, and those colored bands are where we found evidence for QTLs, for wing spots in brown, courtship score in blue, wing display in pink, and body shaking in blue. So each of these traits is determined by one or a handful of major effect QTLs, we think. And there are interesting clusters of QTLs, including right around this yellow gene over on the X chromosome, which has QTLs clustered for all of those traits. And yellow itself is a pigmentation gene, which has already been shown to be involved in the wing spot difference. It's one of a few genes involved in that difference. So we have a paper on this that's just got in press last week, and the next step would be to do more fine-scale mapping on this. We've also done a lot of work in the past on Drosophila melanogaster, which is a cosmopolitan species. It's got to the U.S. from 2 different routes, from West Africa through the Caribbean and also from Europe. And the European flies came out of Africa in an event probably 1,000 or so years ago. There's a lot of interesting diversity, both phenotypic and behavioral, along the east coast of the U.S. So in the east coast, the northern flies tend to be darker, larger bodied, and live longer, and they also have differences in the intensity of their mating behavior. So my former student Roman Yukulevich, who's now a faculty member at Union College, this was his thesis, and he studied a lot of aspects of divergence, including morphology, behavior, and cuticular pheromones. And he demonstrated that there's incipient sexual isolation between U.S. and Caribbean populations. And he, as a postdoc, he went into some of the genetics of sexual isolation in melanogaster, and now he works on a native North American fly system, which also shows incipient speciation. So here's, we've done a little bit of genetics on the pigmentation. So if you do reciprocal crosses that differ only in the source of their X and Y chromosomes, you can show that the X chromosome has a significant effect. So for thorax pigmentation here, so Sudbury is a Canadian line, and 6326 is a laboratory strain that probably came from somewhere in North America. If you measure the luminosity, which is the pigmentation on the thorax luminosity, is the brightness of the thorax, you can see there's a lot of there's a lot of differences among these lines. The autosomes, so we've done some breeding where we've varied the X chromosome and the autosomes. The autosomes don't have a significant effect on pigmentation. It maps actually to the X chromosome. So flies with northern X chromosomes are darker, and this trend is more pronounced in females. I don't have a graph of that. The graph to the upper right is the luminosity by latitude, and you can see that especially in females there's a fairly strong gradient in pigmentation. So high luminosity means low pigmentation. We've also looked at genetic incompatibility and the effects of the X and the autosomes in melanogaster. And this is the big picture here is that there's this pattern, Haldane's rule, which you may have heard of. So when a species hybridization shows different levels of inviability or infertility between the sexes, it's usually the heterogametic or the XY that suffers the stronger effect. So that would be males in mammals and insects, and mammals and fruit flies. It would be females in butterflies and birds. So often you have complete inviability or sterility, such as I mentioned in Drosophila elegans. So several speciation genes in Drosophila species and a couple of other species have been looked at, and we would like to know at what point during speciation these gene effects arise. So we think you need some isolation among the populations, so they would be fixing different alleles that then become incompatible. They're incompatible with each other when they get reintroduced to each other in hybrids. In melanogaster, we can look for these factors by breeding these naturally occurring X chromosomes and autosomes onto different genetic backgrounds, like I did for the stuff I talked about in the previous slide. So when we do that, we've got northern lines, which is sort of north of the Mason-Dixon line there in blue, and we have other lines which would be either 6326, which is a lab line of unknown origin, and then RUM-K, which represents the Caribbean lines that we have. We think it's representative of that. When we put an X chromosome into the autosomal backgrounds, those three different autosomal backgrounds, so the X chromosome from those three places, or with the autosomes from those three places, and we measure the proportion of sterility or lethality, you can see that northern lines in the RUM-K background have a high proportion of sterility and lethality, whereas they don't have a high proportion of in the 6326 lab background or Sudbury, whereas the southern lines represented by RUM-K actually do have a high proportion of sterility in all three backgrounds, including the two backgrounds, non-native backgrounds. So overall, northern X chromosomes are less sterile in the 6326 and Sudbury backgrounds, significantly so, than the RUM-K. So that's incipient. That's evidence for X-linked in X autosome interactions causing incipient reproductive isolation, post-zygotic reproductive isolation. And geographic distance has something to do with that. So these populations show evidence of divergence, and the timescale we're talking about here is probably in the last 500 or so years, because these flies are human commensals. They came to the New World in the age of exploration with humans, probably from Europe, and then through the Caribbean with the slave and other trades that were coming from Africa. So it's an interesting temporal dynamic there, and it's a very recent temporal dynamic. So that's sort of the short end of the time scale. In Drosophila elegans, we can look at a longer end of the time scale. And different strains of Drosophila elegans show much greater divergence than what we see in New World Drosophila melanogaster. So there's some molecular data that we have. So there's an average of 24% species divergence, or the intra versus divided by the interspecific divergence if you use the Drosophila elegans Gunnicola difference. So that we know the Drosophila elegans Gunnicola divergence time is about 2 to 2.8 million years. That's an estimate. And so that would make the divergence between these two elegans strains, Drosophila HK and Drosophila TP. So HK is a strain from Hong Kong, TP is a strain from Taipei, Taiwan, would be about 24% of that overall divergence, which would put it in over 500,000 years to about 780,000 years. So much longer than that divergence among melanogaster strains that we looked at. The pigmentation is different in these two strains. HK is a representative of the more southern brown or more tan strains, which are in mainland Asia and the Philippines and Hong Kong. The TP strain is in the more northern locations, so Taiwan and Japan, and it looks a lot like a melanic mutant in Drosophila melanogaster. When we look at the reciprocal F1 genetics among a lot of different hybrid genotypes between black and brown strains, and we don't have a lot of strains to work with, but we've done this with the ones that we have, we find that if you look at the offspring production per male, there are some combinations that have significantly lower offspring production, group A there, than all the other combinations. And it's dependent on the male genotype. So we've found this asymmetric Haldane's rule effect. So there's lower male offspring production, that's offspring production by males, in genotypes with the HKX chromosome in cytoplasm, and the black strain Y and half of the autosomes. So those two genotypes there. So on that greater time scale, we do find evidence of this Haldane's rule showing up, whereas in the melanogaster incompatibilities, we don't see much evidence for Haldane's rule. It doesn't specifically affect males. So we think we've kind of bracketed this evolutionary of Haldane's rule with two different time points of divergence, and there's a lot of different Drosophila species we can look at with broad geographic distributions with the potential for large geographic isolation or a whole spectrum of geographic isolation. So one of the long-term goals that I have is to look at more of those species and see if we can populate that time spectrum with more snapshots of this process of gradual evolution of the Haldane's rule pattern. Many of you know Rocio Ng. She's been working on the genetic basis of pigmentation differences in melanogaster strains from different parts of the eastern U.S. She used the Drosophila Genome Research Project collection of completely sequenced inbred lines from a population in Raleigh, North Carolina, to look for molecular variation in several pigmentation genes, the ones with red asterisks there on the melanin biosynthesis pathway map. And she's found some really interesting polymorphisms in tyrosine hydroxylase and ebony in particular that seem to be significantly associated with particular pigmentation traits that vary up and down the East Coast. And she was also the first to sequence populations from Maine, Pennsylvania, and to northern and southern Florida to look at whether some of those single nucleotide polymorphisms are in gradients. So we're still sort of doing the data analysis on that work, but it's been really interesting, and it should be a significant advance to go along with a few other stories that have pointed out, in particular in the ebony gene being associated with melanin patterning diversity within species in Africa. So she'll be finishing that up soon, and we should be publishing those papers shortly. And just some newer projects that I've been working on are starting up or thinking about in the last year or two, as a parent, I've really become interested in the evolution of parental care. And there's some areas of this field that have not been well studied theoretically. One of them is just the evolution of parental care from non-care, which has taken place dozens of times in the animal tree of life. So there's a lot of species, even a lot of invertebrates, that have very simple but definite parental care. So we're interested in modeling that using Monte Carlo-based individual genetic models. And I have a more complicated model that applies to a later stage of that process where there's this life history stage that corresponds to the juvenile. And during that life history stage, you have this extended parental care by the parent. And I'm interested in the role of antagonistic pleiotropy. So genes that would be bad for offspring fitness without parental care during that earlier stage that might be really good for fitness once the offspring has matured. It's kind of the reverse of the antagonistic pleiotropy behind aging, which is one of the major evolutionary theories of aging. And then also another life history project I'm interested in is to ask, can sexual dimorphism evolve in a life history stage in which it doesn't appear in nature? And I'm particularly thinking of Drosophila larvae, which we can tell them apart because we can see through their body walls and see their gonads, and the gonads are different. But they, as far as we know, they behave the same as larvae. And I'm really interested in testing whether we could actually select males and females to be different as larvae. There are a few insects that have sexually dimorphic behavior in larvae and sexually dimorphic larvae. It's kind of rare, but it occurs. And the idea is that in Drosophila, and probably in all insects, sex determination is present from the very early embryo, really just about from the zygote. So the cells know whether they're male and or female, and the transcriptional environment is different in those cells. So the potential for expressing genes differently is there at the molecular level. The transcription factors are there. So can you select for, is there genetic variation for selecting for genes, particularly genes underlying behavior, to begin to act differently in males versus females? And I have some ideas of how we can do this using artificial selection in Drosophila. So that's the end of my show and tell. I hope this was interesting. I'm generally around. I don't travel very much these days. And if anybody's interested in this stuff, interested in talking more about this stuff, you can track me down pretty easily, and I'd be glad to talk about it. So sorry about the delay for the second and third year students in getting this, but I hope this is an interesting show and tell.